Hello everyone, today my topic is about xenobiotics. Let's go to the session. Introduction. The xenobiotics are foreign chemical substances that are not naturally produced or expected to be present in an organism's metabolism. These organism compounds can originate from various sources including industrial processes, agriculture, pharmaceuticals, and environmental pollutants. Xenobiotics can be classified based on their chemical or biological properties. Understanding their classification is essential for assessing their potential and impact and developing appropriate strategies for their management. Additionally, xenobiotics can originate from various sources, both natural and synthetic. The first one is the chemical classification of xenobiotics. In this, the organic and inorganic xenobiotics. Organic compounds that are foreign to the biological systems. Example includes pesticides, herbicides, industrial chemicals, pharmaceuticals, and personal care products. Inorganic xenobiotics. Substances that are not naturally occurring in the biological systems. Example includes heavy metals like lead, mercury, arsenic, radioactive elements, and certain industrial pollutants. Next is biological classification of xenobiotics. There are two types. One is endogenous and exogenous. Naturally occurring compounds within an organism's metabolism but present at higher concentration due to external factors. Examples are hormones, bile acids, and other certain metabolic byproducts. Exogenous, like foreign compounds that are not naturally produced by organisms. Example include synthetic chemicals, environmental pollutants, drugs, and xenobiotic introduced through the food and water. Next is the sources of xenobiotics. There are many sources of xenobiotics are there. First one is industrial source. Chemicals used in industrial process, manufacturing and production. Examples include solvents, dyes, and heavy metals from mining operations and pollutants from industrial emissions. Next is agricultural source. Pesticides, herbicides, and fertilizers are used in agriculture. Residues from agricultural practices can contaminate soil, water, and food. Next is pharmaceutical. Active pharmaceutical ingredients and drug metabolites that enter the environment through manufacturing, use, and disposal. Improper disposal of pharmaceuticals can lead to their presence in wastewater and water bodies. Next is environmental pollutants. Pollutants that generated from the human activities such as the combustion process, waste incineration, and vehicle emissions. Examples are polycyclic aromatic hydrocarbons (PAH), volatile organic compounds, and persistent organic compounds. Pollutants. Natural sources. Naturally occurring compounds that can act as xenobiotics in certain contents. Examples like plant secondary metabolites, mycotoxins, and toxins produced by the marine organisms. Metabolism of xenobiotics. Xenobiotics undergo various metabolic transformations within an organism, leading to the biotransformation and elimination. The metabolism of xenobiotics includes a series of enzymatic reactions that occur in two main phases. One is phase 1 and phase 2 metabolism. This is the overall structure. This is the overall structure of the overall process of the metabolism of xenobiotics. Phase 1 metabolism involves the initial biotransformation of xenobiotics primarily through oxidation, reduction, hydrolysis reaction. This phase aims to introduce or unmask functional groups on the xenobiotic compound, making it more amenable to conjugation reaction in phase 2. The key features of the phase 1 metabolism include oxidation, reduction, hydrolysis. So, high oxidation. Cytochrome P450 enzymes are the major catalyst involved in xenobiotic oxidation reaction. CYP450 enzymes introduce hydroxyl, carboxyl, or epioxide groups into the xenobiotic molecules. Oxidation reaction enhance the water solubility of xenobiotics, making them more susceptible to subsequent conjugation reaction. Next is reduction. Reduction reaction involves the addition of electrons to the xenobiotic molecule, usually mediated by reductase enzyme. Reductive reaction can lead to the formation of more polar or less toxic metabolites. Next is hydrolysis. Hydrolytic reactions involves the cleavage of chemical bonds through the addition of water molecules. xenobiotics such as esters, amides, epoxides can undergo a hydrolytic reaction resulting in the formation of hydrophilic metabolites. Next is phase 2 metabolism. It involves the conjugation of phase 1 metabolites with the endogenous compounds such as glucuronic acid, sulfate, lysine, or glutamine. This conjugation process further increases the water solubility of xenobiotics, facilitating their elimination from the body. Key features of phase 2 metabolites include glucuronidation, sulfation, other conjugation reactions. The glucuronidation involves the transfer of glucuronic acid from UDP glucuronic acid to a xenobiotic or its phase 1 metabolite. Glucuronidation reactions are catalyzed by UDP gluconyl transferase enzyme. Glucuronidation is a common pathway for the metabolism of many drugs, environmental pollutants, and endogenous compounds. Sulfation involves the transfer of sulfate group from the 3-phosphoadenosine 5-phosphosulfate to a xenobiotic or its phase 1 metabolite. Sulfation reaction are catalyzed by sulfotransferase enzyme. Sulfation is an important pathway for the metabolism of phenols, amines, and certain drugs. Other conjugation reactions. Other phase 2 conjugation reactions include amino acid conjugation, acetylation, methylation, glutathione conjugation. These reactions add functional groups to the xenobiotic, enhancing their water solubility and facilitating excretion. Next is biodegradation. Biodegradation refers to the process by which microorganisms utilize xenobiotics as a source of energy and carbon, leading to the transformation and eventually mineralization. Microbial biodegradation of xenobiotics plays a crucial role in environmental remediation, wastewater treatment, and sustainable management of contaminants. Bioremediation. Bioremediation utilizes microorganisms and their metabolic activities to degrade and detoxify xenobiotics in contaminated environments. In situ biodegradation involves the use of native microorganisms, while ex situ biodegradation occurs in controlled laboratory or industrial settings. Environmental applications such as biodegradation can be applied to address communication from various sources, including petroleum, hydrocarbons, polycyclic aromatic hydrocarbons, chlorinated compounds, and pesticides. Biodegradation can be conducted in soil, water, sediments, and even air, depending on the specific contamination scenario. Enhancement strategies like biodegradation can be enhanced through strategies such as bioaugmentation, introduction of specific degrading microorganisms, biostimulation, provide necessary nutrients and conditions for microbial growth, and bioventing, provide oxygen for aerobic degradation. Next is bioremediation. Bioremediation is a powerful approach that utilizes living organisms or their compounds to degrade or transform xenobiotics into less harmful or non-toxic substance. It offers a cost-effective, environmental-friendly, and sustainable method for remediating contaminated sites. The first one is petroleum hydrocarbons. Microorganisms can degrade hydrocarbons found in the crude oil spills, petroleum contaminated soils, and groundwaters. Hydrocarbons degrading bacteria such as Pseudomonas, Al- Alcanvivoras, and Rhodococcus. Chlorinated compounds. Certain microorganisms have the ability to degrade chlorinated solvents, pesticides, and other chlorinated compounds. Dihalococcosidae species are well known for its ability to dechlorinate compounds like trichloroethylene and polychlorinated biphenyls. Next is heavy metals. Microorganisms can also play a role in bioremediation of heavy metals by transforming or immobilizing them. Some bacteria and fungi can produce metal-binding compounds or enzymes that facilitate metal sequestration or conversion into less toxic forms. Next is biocatalyst. Biocatalyst involves the use of enzymes or whole cell as a catalyst to facilitate chemical reactions. In the context of xenobiotics, biocatalyst offers a promising approach for the transformation, detoxification, and synthesis of these compounds. The first one is pharmaceutical industry. Biocatalyst plays a significant role in pharmaceutical synthesis, particularly in the production of chiral drug compounds. Enzymes such as oxidoreductase, hydroxylase, transferase are employed to introduce specific functional groups or stereochemistry. Environmental applications. Biocatalyst offers potential solution for the detoxification and degradation of environmental pollutants, including pesticides, herbicides, and industrial chemicals. Enzymes can be employed to enhance the breakdown of persistent organic pollutants and facilitate their removal from the environment. Fine chemicals and specialty chemicals, biocatalytic processes are increasingly utilized for the synthesis of fine chemicals and specialty compounds. Enzymes enable the production of complex intermediates and final products with a high selectivity and efficiency. Thank you so much for this wonderful opportunity. Thank you.